Assalamu alaikum students, welcome to my YouTube channel with new lecture on signaling molecules. In previous lectures we discussed about the communication of bacteria, scientifically called as quorum-sensing. So in this lecture we will discuss briefly about signaling molecules, their types and how these sensors work for bacteria in communication purpose. So the signal molecules, these are the small chemical molecules also called as autoinducers utilized by the bacteria for the communication purpose. Several types of the quorum-sensing signal molecules have been reported in bacteria. For example, N-acyl homoserine lactone, autoinducer peptides, autoinducer 2, gamma butyrolactones, furanosyl borate diesters and quinolones. These all are the different types of the chemicals that are produced and utilized by the different types of the bacteria as the quorum sensing signal molecules. Major quorum sensing molecules. Among the above discussed quorum sensing molecules, some of these are known as major quorum sensing molecules that are widely produced by the bacteria. These are the N-acyl homoserine lactones, autoinducer peptides and autoinducer 2. Autoinducer peptides, these are the specific quorum sensing molecules that are produced by the gram-positive bacteria. If we talk about the autoinducer 2, these are the specific type of the quorum sensing molecules that are produced by both gram-negative and gram-positive bacteria. It means these are the signal molecules that can be produced and utilized together by both of the bacterial groups. N-acyl homoserine lactones. We will discuss in detail about these type of the quorum sensing molecules that are widely produced by large numbers of the pathogenic bacteria, and they use the type of the signal molecules to communicate with each other and to cause the pathogenicity. Examples of the N-acyl homoserine lactone. N-acyl homoserine lactone have been reported from carbon 4 to carbon 18. For example, C4HSL, 3-hydroxyl group of the C4HSL, C6HSL, oxo group of the C6HSL, C8HSL, 3-oxo C8HSL. In this way, there are several other types of the N-acyl homoserine lactone. A single bacterium can produce more than one AHL. Even in some reports, one bacteria can produce more than 5 to 6 N-acyl homoserine lactones, and these all the produced signal molecules will be utilized for the communication purpose. Detection and identification of the N-acyl homoserine lactone based signal molecules. How can we detect and identify these quorum sensing signal molecules produced by the bacteria? So the biosensors, what are the biosensors? The biosensors, these are the specific type of the bacteria. They don't produce the quorum sensing molecules themselves. However, they have an ability to sense, to detect the quorum sensing molecules that are produced by the other bacteria. These biosensors are also called as the reporter strands. This detection method will be applied through the cross-feeding bioassay as well as well diffusion bioassay. After the confirmation detection by the biosensors, the N-acyl homoserine lactone quorum sensing molecules, they can be identified through the RP-TLC, reverse phase thin layer chromatography, GCMS and also HPLC. Examples of the biosensor bacterial strains. Among these, two strains are mostly used to detect the N-acyl homoserine lactone-based quorum-sensing molecules. One is Agrobacterium tumefaciens strain A136 and other one is the Quorum-bacterium violacem CV026. When there is any production of the signal molecules by a test bacteria, the A136 will produce the purple blue color. However, CV026 will produce the violacine color in the presence of the signal molecules. Control of the multi-drug resistant bacteria. There is one question raised by many students that how can we control the disease causing bacteria which have become resistant almost all types of the antibiotics. So this is a very interesting and informative question, since it is a fact that majority of the pathogenic bacteria now become resistant against broad range of the antibiotics, and these all are called as the multi-drug resistant bacteria. Unfortunately, the resistant rate increased very high after the outbreak of the COVID-19, maybe due to misuse of the antibiotics. So research is underway and several research articles have been published to overcome antibiotic resistant bacteria by disrupting the quorum sensing system, as it is now clear that most of the multi-drug resistant bacteria use the quorum sensing system to cause the pathogenicity disease. So to control or overcome the quorum-sensing system in pathogenic bacteria, the process is called as the quorum-quenching, in which certain enzymes or inhibitors are used as a quorum-sensing inhibitors which destroy the quorum-sensing system, and in result there is a prevention of the pathogenicity or disease. You can see the quorum-sensing bacteria in which the inhibitors, if they are inserted, there is a destruction of the quorum sensing system, and in result there is no any pathogenicity or disease.